Welcome to our channel Botany with YouTube. Today I am here, a PhD scholar. I am going to present a topic, seed germination. What is seed germination? Seed germination may be defined as the fundamental process by which different plant species grow from a single seed into a plant. This process influences both crop yield and quality. A common example of seed germination is the sprouting of a seedling from a seed of an angiosperm or gymnosperms. Germination is emergence of normal seedling from the seeds under ideal condition of light, temperature, moisture, oxygen and nutrients. Germination is emergence of radicle and plumule through the seed coat. Botany with YouTube is our channel. Try to subscribe our channel firstly. Step of seed germination. Phase 1, imbibition. The dry seed take up water rapidly by the process of imbibition. It is the first step that moves the seed from a dry, dormant organism to the resumption of embryo growth. The extent to which water imbibition occurs is dependent on three factors: consumption of seed, seed coat, permeability, water availability. The initial rapid uptake of water by the dry seed during phase 1 is referred to as imbibition. Water potential gradient drive water uptake in both cases. The causes of gradient is different. It starts the growth process by activation of enzymes. You can see here the process of imbibition. Phase 2, lag phase. The rate of water uptake slows down until the water potential gradient is restabilized. Phase 2 can thus be thought as lag phase preceding growth, during which the solute potential of the embryo gradually becomes more negative due to the breakdown of stored food. Stored volume may increase as a result of rupturing of seed coat. By rupturing of seed coat, radicle emerges to form a primary root. The seed starts absorbing underground water. After the emerging of the radicle and the plumule, shoots start growing upward. You can see here lag phase in diagram. Mobilization of reserve food is the third phase. Water uptake resumes due to the decrease in the seedling growth. The stored food reserve of the seed are fully mobilized. During phase 3, the rate of water uptake increases rapidly due to the one of the cell wall loosening and cell expansion. Thus, the water potential gradient in phase 3 embryos is maintained by both cell wall relaxation. In the final stage of seed germination, the cell of seed become metabolically active, elongates and divide to give rise the seedling. Seed germination process you can see here in diagram. Radicle, hypocotyl, seed coat, cotyledon, hypocotyl, epicotyl, cotyledon, first true leaves. Physiology of seed. Structure of seed. Seed coat, micropyle, helium, cotyledon, radicle, plumule, endosperm. Structure of seed. Seed coat. The outer covering of seed is called seed coat. It protects the embryo from entry. Micropyle. It is a tiny pore in the tester that lie on the opposite of the tip of the radicle. It permits water to enter the embryo before active germination. Hilum. It is a scale left by the stalk which attach the ovule to the ovary wall before it becomes a seed. Radicle. This is the embryonic root. It's usually first part of the embryo to push its away out of the seed during germination. Plumule. This is the embryonic shoot. It appears as bud and gives rise a shoot. Cotyledon and endosperm. You can see here in diagram an embryonic leaf in seed-bearing plant, one or more of which are the first leaf to appear from germinating seed. Food storage tissues, result of double fertilization. 2/3 genome of the maternal origin. The triploid endosperm is composed of 2 tissues, starchy endosperm. Botany with YouTube is our channel. Share our channel, please. Requirement for seed germination. Requirements: abiotic factors, gases, water, temperature, light. Biotic factors: seed dormancy. Gases: air is composed of about 20% nitrogen, 0.03% carbon dioxide. About 80% of nitrogen gas. Oxygen is required for germination of most species. Carbon dioxide concentration higher than 0.03% retard germination, while nitrogen gas has no influences. Temperature. Seed germination is a complex process involving many individuals, reactions and phases, each of which is affected by temperature. The optimum temperature for most seed is being between 15 and 30. The maximum temperature for most species is between 30 to 40 centigrade. The response to temperature depends on a number of factors including the species, variety, region, quality of the seed and the duration of time for harvest. Water. Water is the most essential factor. The water content of mature air-dried seed is the range of 5 to 15%, well below the threshold required for fully active metabolism. In addition, water uptake is needed to generate the turgor pressure that powers cell expansion, the basis of vegetative growth and development. Light. Generally, seed requires darkness to germinate, but sometimes it requires light. Photoblastism or photoblastic. Seed responds to light for germination, named as positive photoblastism and negative photoblastic. Phytochrome is a plant pigment found in cytoplasm that senses the presence of red light. Seed dormancy. This is a condition in which the seed are prevented from germinating even under the favorable condition. Some seed mature in the fruit but do not germinate until released from the fruit. This type of dormancy cannot be overcome. It can only recover by waiting. A seed may require certain amount of light to germinate, causing the seed to remain dormant until exposed to light. This type of dormancy can be recovered by scarification. Botany with YouTube is our channel. Like our channel, please. Types of germination. hypogeal germination, apigeal germination, and viviparous germination. In this germination process, apigeal, in this the cotyledons are raised out of the soil and generally become green and photosynthetic. In dicot, they are pushed up and rapid extension of hypocotyl before growth of epicotyl. Apigeal germination occur in beans, castor, and sunflowers. Hypogeal germination, you can see here in diagram. In this type of germination, the cotyledon remain underground. Hypocotyl growth is restricted. The epicotyl grows to raise the first leaf out of the soil. It occurs in dicotyledonous seed of gram, pea, mango, and in monocotyledon like rice, maize, wheat. In this type of germination, this is a special type of germination occurring in mangrove plants. These plants generally grow in salty lakes, sea coast, and deltas. The embryo emerges out of the fruit with a massive radicle pointing downward. Thanks to visit our channel. Allah Hafiz.